SARS-CoV-2 virus infects host cells through the recognition of spike protein, ACE2 receptor, inducing virus and host membrane fusion, or virion endocytosis, and followed by uncoating, genome duplication, virion assembly and egress. The E are Golgi intermediate compartment. ERGIC is just the place where virions are assembled by structure proteins such as spike N, M and E. The proper ERGIC localization of host proteins depend on vesicle trafficking system in our body. COP2 vesicles mediate the transport of proteins from the ER to the Golgi complex through ERGIC, while COP1 vesicles are mainly responsible for the retrograde trafficking from the Golgi complex or ERGIC to ER. SARS-CoV-2 hijacks our COP1 and COP2 protein trafficking machinery. For an instance, ERGIC localization of SARS-CoV-2 spike relies on the dynamic COP1 and COP2 trafficking to facilitate virion budding here. Proteins can be segregated by different vesicles depending on the specific sorting signal recognized by distinctive trafficking machinery. SARS-CoV-2 spike binds to both the COP1 and COP2 coats with COP1 sorted KLHYT motif and COP2 DSE motif in spike cytosolic tail, respectively. The trafficking balance between COP1 and COP2 vesicle transporting helps spike to be localized in ERGIC. Based on the structural features of SARS-CoV-2 spike bound to COP1 coat and the biochemical characterization of this interaction, our team designed a peptide drug named TAT-WDM, which can enter cells efficiently with a much stronger affinity to COP1 than spike. TAT-WDM is able to compete SARS-CoV-2 spike interaction with COP1 in vitro and in cells. The attenuation of spike COP1 trafficking by TAT-WDM promotes more SARS-CoV-2 spike host cell membrane trafficking, since spike is often the target of anti-COVID-19 antibody therapy. TAT-WDM is capable of assisting infected host clearance via exposing more spike in host cell surface. The increased distribution of spike on the surface of cell membrane is helpful for their recognition and binding of spike-targeting antibodies, and then promotes the recruitment of NK cells by antibodies through Fc receptors to enhance the ADCC effect and improve cytotoxicity of NK cells on target cells. In brief, TAT-WDM could significantly promote the antibody-mediated clearance of virus-infected cells. In this study, we have invented a COP1 sorting inhibitor that promotes SARS-CoV-2 spike cell surface trafficking. In application, TAT-WDM promotes antibody-mediated Fc effector function to eliminate infected cells and inhibit virus spread. Further, their surface distribution of spike favors spike mRNA vaccine-induced B-cell activation, and thus is able to be used as an adjuvant for spike mRNA vaccines.